We are going to talk about the formation of proteins using peptide bonds and amino acids. The amine groups will be represented by the blue molecules shown here. The R groups will be represented by the green molecules. The carboxyl group, shown here, is especially important in the formation of peptide bonds. When forming a peptide bond between two amino acids, the oxygen and the hydrogen are lost from the carboxyl group, and they form a water molecule with a hydrogen that is lost from a neighboring nitrogen bonded to an amino acid. This will form a water molecule, and then a covalent bond is formed between that carbon and nitrogen. This covalent bond is referred to as a peptide bond, which can be seen as these orange bonds here. These peptide bonds are restricted in their rotation due to the resonance that can be created because of these neighboring double bonded oxygens to carbons or carbonyls. This leaves the rotation restricted to the alpha carbon. The alpha carbon is a carbon that is attached to an amino group, a carboxyl group, and an R group. The alpha carbon has rotation around between the nitrogen and the alpha carbon, and this is called the phi angle of rotation. The angle of rotation between the carbonyl carbon, or the carboxyl group in this case, is the psi angle of rotation. So the phi angles of rotation can be seen as the pink bonds here. These are the phi angles. And the psi angles of rotation can be seen as the green bonds between either a carboxyl group or a carbonyl and an alpha carbon here. We are now going to discuss the secondary structures of the polypeptide chain. As the structure sits right now, it is in the beta strand conformation. In the beta strand conformation, it is most commonly found that the phi and psi angles are at -120 and 120 degrees. If we wanted to adjust this into an alpha helix structure, we would need to rotate the angles into -60, -60 degrees, keeping in mind that we cannot rotate around the orange peptide bonds. So we are now going to rotate this beta strand structure into an alpha helix structure. After rotating the bond angles to reach -60, -60 phi and psi angles, we can now see that a helix shape is starting to form in the molecule. So we have now created the alpha helix form of this polypeptide chain.